Good day and welcome, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Menarini Silicon Biosystems, I'd like to welcome you to CLIA validation of DEPArray and its ability to overcome limitations associated with current oncological techniques. My name is Elizabeth Lamb, and I'll be the host for today's event. At this time, I'd like to introduce our moderator for today. She is Dr. Faraday Bischoff, PhD, Chief Clinical Development Officer for North America Menarini Silicon Biosystems. Welcome, Dr. Bischoff. The presenter ball is yours.
Thank you, Elizabeth. Good morning, everyone, and thank you for joining us today for our webinar presentation entitled CLIA Validation of the DEPArray and its Ability to Overcome Limitations Associated with Current Oncological Techniques. I'll begin with a very brief overview addressing the unmet need, followed by presentations from my colleagues, Drs. Niccolo Menarisi, Chief Scientific Officer of Menarini Silicon Biosystems, and Dr. Matthew Moore, who's Principal and Co-founder at ResearchDx. In the practice of precision medicine, we have a moderate number of companion diagnostics which are coupled with treatment decisions to a measurable biomarker. So this is really intended to guide physicians in selecting the right therapy for the right patient at the right time. Each of these biomarker assays, as they're described on the FDA website, is dependent on a tumor tissue biopsy. This biopsy is in the form of an FFPE sample that is then utilized to perform the various downstream applications that are highlighted on this slide, including fluorescence in situ hybridization, or FISH, NGS for next-generation sequencing, immunohistochemical staining, and more recently, analysis of the immune-infiltrating lymphocytes. However, there are appreciable limitations that can impact the utility of FFPE specimens in the clinical setting. These limitations fall into basically two categories. The first being pre-analytical challenges associated with samples based on where the biopsy is obtained from, how the biopsy is performed, and as well as how the sample is processed, fixed, and stored for subsequent use. Secondly, there are analytical challenges. These challenges really fall into specific categories, most of which involve heterogeneity at the intracellular or intragenomic levels. As a result of these limitations, the noted downstream assays as applied to FFPE specimens can lead to test failure or inconclusive results, thus leaving a significant percentage of cases without genomic biomarker information that's needed in order to make that clinical decision as to what therapy to select, whether selection of therapy needs to be changed, altered, or combined with other downstream treatments. When we consider NGS, for example, the leading cause of failure is insufficient tissue or DNA for that downstream assay. And as a result, many turn to CTCs as an alternative, particularly when tissue is either inadequate or insufficient for that downstream analysis. Circulating tumor cells offers the advantage of being not only a surrogate for tissue biopsy, but also provides potentially a tool for early evidence or detection of resistance or recurrence of disease. Furthermore, unlike tissue biopsy, liquid biopsy in the form of CTCs is repeatable throughout the course of treatment, can be used not only for enumeration in terms of prognostic valuation, but also to determine the comprehensive genomic characteristics associated with those cells or tumor tissue. However, as we all would appreciate, CTCs are heterogeneous, and this, in fact, can cause difficulties in determining clinical utility or performing the needed validation studies downstream. Therefore, the DEPArray offers a solution that can improve not only the utility of tissue biopsy specimens, but also liquid biopsy specimens in the form of CTCs, by overcoming the general challenges associated with there being insufficient material, where we can now recover those few cells that are present and utilize those cells for the downstream analysis. We can stratify these cells based on morphology, immunofluorescence, phenotype, and/or DNA content. But very importantly, we can now consider standardization of the input material for our downstream assays based on the DEPArray platform. So with that, I will now hand over the presentation to Niccolò Menarisi, who will present to you the DEPArray and Ampli1 workflow for CTC isolation and molecular characterization. Niccolò?
Okay, thank you, Faride, for the introduction, and welcome everybody. So I will start my presentation looking at the workflow, especially to the core of our technology, which is the DEPArray. And the DEPArray NXT in particular, this is the third generation of our proprietary technology. It's a benchtop version, and it's a system composed of the machine software specific to run in a user-friendly format, sorting of cells, which occurs in disposable cartridges, which have at their core a microelectronic chip. And this is what you see in the center of the green, let's say, cartridge. That's the core of our technology. It's an array of electrodes, over 300,000 electrodes, which create over 30,000 cages. And the system affords image-based digital sorting. So all the cells are trapped in stable levitation in the 30,000 cages. We recognize the cells, and then based on images and scatter plots, we can recover up to 96 single cells, or pool of cells up to 500 cells in a single pool, delivering 100% pure cells at the output. It's also very gentle with live cells, and importantly compatible with small cell loads, which is what you get in many clinical samples, like fine needle aspirates or enriched CTCs from blood. And in particular, focusing on liquid biopsy and circulating tumor cells, we really have now the possibility to frame our technology and put it to work in the CTC field, turning what was a prognostic indicator, very strong, independent indicator of patient survival. The Kaplan-Meier curves, which you see, are based on the studies of the cell search technology, which Menarini acquired, as you may have seen from the news. And using this, which is the only FDA-approved and best-in-class technology for CTC isolation, which really opened up the field, what we have the possibility to do is now to turn those CTCs, isolate them, and turn them into a source of predictive biomarkers in order to stratify patients with the aim of understanding the genetic background of each patient tumor and possibly address the right therapy to the right patient. So how do we do this? So our aim is to really deliver sample to results turnkey solutions, and we have worked SOPs, which Matthew will present as regard to the CLIA validation of these procedures. Really from blood, we can use different enrichment technologies. Eventually, you go from 1 in a billion to 1 in 1,000. And of course, the cell search is the election method as of today, but we are compatible with our DEPArray with the different technologies. And then you go for the DEPArray to go for single cells. And then we offer a number of kits and regions for the downstream, because in order to solve also the challenge of analyzing the genetic makeup, the genome and transcriptome of individual cells. So doing whole genome amplification with our Ampli1 product line, and also providing library preparation kits for low-pass whole genome sequencing, for copy number profiling, or targeted sequencing with our Ampli1 OncoSeek. So, and this is really a change of paradigm, and really is starting to bring very important fruits, because with this this workflow, this is the header of a recent publication in Nature Medicine, whereby a group in Manchester, the group of Caroline Dyer and Jeb Brady in Manchester, showed that by analyzing the copy number profiles of CTCs in small cell lung cancer, they were able to discover a signature which is able to predict whether a patient is chemosensitive or chemorefractory. And indeed, this was done with a really very high accuracy. They did first a training set with a sensitive and refractory patient. They came up with a signature which was 100% accurate, and then they validated this signature on a test set in a prospective manner, and still retaining a very high accuracy, overall 90%, and more than 83% for the prospective cohort. So the advantage here is that while, as you see in the Kaplan-Meier curve on the left, if you just give the drug, you need to wait 90 days before knowing how will the patient respond, and whether that specific patient will be on the green line, so with better outcome, better overall survival, or maybe whether the patient will be chemorefractory and have a poor outcome very quickly. If you are able to do this analysis, now you, at the baseline, you can predict in advance whether one specific patient belongs to which cohort. So if it belongs to the chemorefractory, of course, there will be the idea to find something else. And this is, of course, subject to future studies, but this is a good example of the power of this kind of analysis at the single-cell level. And indeed, the single-cell resolution, according to the results reported by this group, is really coming up as something to look at. Because what they found was that if you look at, across the patients which had CTCs, all calling the patient as chemorefractory or chemoresponsive, the accuracy was above 90%, also in the prospective cohort. But if certain CTCs were saying, okay, the patient is refractory, but others claim that the patient is responsive, so there is discordance between single CTCs, then the accuracy was lower. So this is something very interesting to look at. And in order to really make the most out of this kind of analysis, we actually came up with the idea to streamline this kind of analysis by using the specific properties of Ampli1 whole genome amplification. And in the conventional methods, to do a low-pass whole genome sequencing, you need to have a number of steps, enzymatic and fragmentation by sonication, so like digestion of the adapters, ligations, before getting to PCR amplification, pooling, and then sequencing. With our new kit on the Ampli1 LowPass protocol, exploiting the deterministic adapter of Ampli1 WGA and deterministic digestion, we were able to come up with a very simple protocol where you just do a PCR for each individual sample, then pull everything, and then you can go directly after a site selection to the sequencer. And what we found is that with this protocol, we were actually able to get at the single cell level a very specific and sensitive and also quantitative assay. Because on the top panel here, what you see is, for example, a normal leukocyte, which has 2 copies across all the autosomes from 1 to chromosome 1 to 22, as you see here. Whereas on the bottom panel, you see a cytokeratin positive, cytokeratin is in green, CD45 negative, and DAPI positive circulating tumor cell. And what you get is then a profile with aberrations, amplification, and losses very, very clear, and by which you can also assign integer copy numbers to each region of the genome. So this is extremely convenient because it's a simple PCR and cost-effective, and also very flexible because you can adjust the resolution whether you want to have more or less resolution. You just put more or less reads in your sequencer for any given sample. And it's very also robust because it's resilient to DNA degradation, this approach, and it really lends itself very well to make a quantitative analysis of the copy number profiles. So all in all, this is something which we are really able to apply in patients. And this is, for example, a case study, an example, where from a lung adenocarcinoma patient, we have isolated CTCs through our workflow, including cell search, DEPArray, and Ampli1. And using this Ampli1 Low Pass approach, we have analyzed 4 CTCs, and you see here that in this case, the CTCs show remarkable differences. And in fact, apart from chromosome 7 amplification, there is little in common between the other CTCs, except that 3 of them have an amplification on chromosome 13, but another one has not that amplification. So there is indeed the need to go to this ultimate resolution if you wanted to understand the genetic makeup of these cells. And what's even more exciting is that this kind of approach is really very information-rich, and looks like from recent findings from other groups that the tumor aneuploidy also has to do not only with chemotherapy, like the group of Manchester has shown, but also to immune oncology response. Because what they found was that the tumor aneuploidy was inversely correlated with the patient survival and response to immune checkpoint inhibitors. This is all very promising, and we have now this Ampli1 Low Pass kit available on the market for the Ion Torrent platform and for our fee-for-service customers. And in less than 1 month from now, we will also launch the Illumina version with up to 96 barcodes. So all in all, this is really the ultimate step into digitizing. So not only digital sorting, but digital resolution at the single cell level in order to call copy number changes across the genome. And this I like to make it the comparison with the kind of barcode, which is unique for each cell, very information rich. And as we have seen from the results before, these copy number aberrations look like they are very intimately linked to the tumor biology. So we are very optimistic about the potential of this technology to reveal new biomarkers, and we think we will increasingly see it used in the clinic. But of course, we are not only about this. We also have aware that single gene and gene sequence variants and focal copy number changes are important information in the clinics today. And so we're actually working also for launching an OncoSeek panel for Ampli1, which is about getting information on mutations in about 60 genes, including copy number changes. And we published in 2015 about the first result from the prototype of this technology, which we are going to launch, and combining cytokeratin positive cells analysis with the low pass and somatic variant investigation, as you can see here from this panel, where in the bars the light blue indicates the variant frequency for each cell, and as you can see in the bottom panel, there are no somatic mutations. This is the white blood cell, which is normal, whereas we find, for example, in this patient, heterozygous SLT3 mutation and a P53 mutation homozygous in one cell but not present in the other. So we will be able to get all this information, including copy number changes, because when you look at single cell, then it becomes easier also to recognize whether you have an amplification. This is a preview of our prototype kit, and this is a single SKBR3 cell amplified with whole genome amplification. And what you see here is the fold change for the 19 genes which we investigate for copy number aberrations. And the white blood cells are represented by blue bars, and they cluster around the one fold. So they're all normalized and come across to the normal value. But we notice in the single SKBR3 cell line that you find very prominent HER2 amplification along with the EGFR and MYC copy number codes. So this is about liquid biopsy and single cells. But of course, we have the potential to work and make a difference also for low tumor cellularity FFPE samples. So in this case, the problem is less extreme. You don't start with 1 in a billion, but still, if you start from, let's say, 5%, these samples are very problematic for NGS. And indeed, this represents a real clinical problem. Recent news from the early basket trials revealed that when you actually try to implement precision medicine, about 20%, 1 in 5 of patients do not have enough malignant cells in their biopsy, either because the tumor cellularity is too low or because the overall amount of material is insufficient. And to address this problem, we use the power of digital sorting with DEPArray, and we published last year in Nature Scientific Reports. You can find this publication. It's open access. You just Google digital sorting FFPE and it pops up. And we published that we can use DEPArray to purify 100% at 100% purity cells from FFPE samples. And this again make a difference and we can build now a workflow for precision medicine, starting from even very small tumor biopsies. We have protocols and kits, the FFPE-QC kit to assess the DNA integrity, and the FFPE sample prep kit, which is the next in line to stain and suspend the cells from the sample in order to load it as a cell suspension on the DEPArray. And from there, we isolate pure cells, we lyse the cells, and we can prepare a library for targeted sequencing with what we call the DEPArray OncoSeek panel, or library preparation for general large-scale investigation like low pass or whole genome or whole genome sequencing or whole exome. So in this case, what we are able to do, thanks to DEPArray, is to address some of the key bottlenecks for implementation, ubiquitous implementation of precision medicine in every patient. The first one being tumor content. And if we look around, for example, for foundation medicine, the specification is that you need at least 30% tumor content. Or if you are going for a non-combined focus assay, which includes copy number aberration code, the bar is even higher because you don't want your copy number to be diluted by normal cells, and so you require 50% purity. So with DEPArray purification, we can relax the constraint on the input sample tumor content down to 5% or even below. Actually, we didn't add samples with less than 5%, but this is what we know we can work with. And the second point is that even the total amount of cells beyond the frequency of tumor content is also a key requirement. And again, thanks to our workflow, we can really basically go at about 1/4 or even 1/10 of what's possible with foundation medicine specification in terms of tissue volume. This is what we are currently validating in our CLIA validation effort with the group of Matthew Moore, which we'll speak later. And also, if you reason in terms of DNA input, thanks to the fact that we have pure sample, we can relax and go from 10 nanogram specification of the Oncomine down to sub-nanogram input. And we can do this because we don't mash up everything and then try to make sense of the mixture, which is basically very likely to lead to inconclusive and noisy results. But we take a bold step in the pre-analytical phase, and we separate the various different components of the tumor microenvironment. So cancer cell on one side, stromal cells, and also possibly immune infiltrate of a given sample. And as we do this, we look at with different eyes to our sample. So after disaggregation, you find by immunofluorescent markers, by 19 and keratin, the different populations. In this example, you have the stromal population by 19 positive and keratin negative, and you find the tumor cells in the keratin positive and by 19 negative population. As well as you can find a heterogeneous cluster and tell them apart from real double positive cells by looking at the images as enabled by the DEPArray and looking at the co-localization of the fluorophores. And in addition, we can also stratify and segregate cells based on their DNA content, as we use the stromal population as an internal control, thanks to the peak of diploid, which you see here, the peak of diploid DNA from by 19 positive cells. And so here we can see the tumor population, which has a shift in this specific patient of DNA, which is the hallmark of an aneuploidy in the tumor population. So we can use this further step to segregate the sample. So then we get to pure cells, and what we can do is, for example, if you have a specific clinical question for a number of genes which are included in typical cancer analysis, you have we can offer the DEPArray OncoSeek panel, which is one of the kits which we are validating currently in the CLIA. This is a relatively, let's say, standard design, the usual suspects, except that beyond sequence variants, since we have a pure sample, we can look at copy number alterations as well. And most of all, with this kit, we made a big effort to be compatible with very minute amounts of inputs. We are talking about 100 to 300 FFPE cells, depending on the quality of the DNA. And this corresponds to really down to sub-nanogram quantity of DNA, which is basically 1 order of magnitude to what all other kits available on the market can reach. And this is a streamlined single tube 4-hour protocol for the Illumina platform. And I'm going to show you some example with starting with this pancreatic patient sample. with a very low tumor cellularity estimated by the pathologist between 10 and 20%. And we have made several replicate recoveries for stromal and tumor, and also for the original unsorted population. And as you can see here, in basically what we have for the variants which we have found, we have a nice separation between stromal and tumor population. For example, KRAS is clearly an heterozygous, 50% variant present only in the tumor and absent in the stromal population. Whereas for the P53 mutation, we find that it's homozygous mutant, so about 100% in the tumor and 0 in the stromal. And also we see lots of heterozygosis in form of germline SNPs, which are 50% in the stromal, and they flip to either 0% or 100% variant in the tumor, depending on whether you have whether the tumor has lost the wild type or the variant allele. And all of this is really difficult to gauge from the unsorted population, where numbers are all over the places, and it's very, very difficult to make sense and to understand the underlying biology. And this is also true if you are going for the copy number. This is a breast cancer FFPE sample with 10% tumor cellularity, and we show here for the HER2 gene the fold change which you observe if you collect multiple stromal population. They cluster around the normal value, normalize it to one fold change, so 2 copies. When you have a sorted a tumor pure, you clearly see loud and clear. This is down to 75 cells with very significant increase in copy number changes. And interestingly, due to the dilution of normal cell in the unsorted population, these 2 replicates don't make it above the thresholds of 2-fold change. So 2-fold from 2 would be 4 copies, which is the threshold to call a patient HER2 amplified, very importantly. So all in all, this is for focal and targeted sequencing, but we also offered DEPArray LibPrep to enable large-scale analysis. And this is, of course, is then for Illumina platform. And again, this library prep kit has been designed for minute amount of input, 200, 300 FFPE cells, so 1 to 2 nanograms, and enables low-pass whole genome sequencing, whole exome sequencing, or even whole genome sequencing. And I'm going to show you some example. What does it mean to purify? So when you have a sample unsorted, you can somehow see some changes, some deviations. But again, this is a relatively low tumor content, so you can basically see that there are some deviations here, but the software doesn't call them, except if there are very high amplification, it's able to detect them. But in practice, what you have, that this is the sum of a lot of this diploid normal with a tiny amount of the real tumor, which is, when it's 100% pure, you can really assign to, for example, here 3 copies, 2 copies, 4 copies here, or 1 copy here. And really, when it was very well detectable here, it was really out of the roof in the sorted population. So this is really the difference in having a pure sample. And also, if you go for sequence variant, dividing the tumor and stromal content is key. This is an example where we plot the D allele frequency across the whole exome of the germline heterozygous SNPs of this pancreatic patient sample. And in the unsorted population, it's basically all across the 50% threshold. But if we separate the stromal and the tumor content, clearly it comes up that the tumor has lots of heterozygosis, and also, as you can see here, with the doubling bands. This is the hallmark of a copy number change. And putting together all the information, the low pass and the whole exome, you can really understand with exquisite details the genetic makeup of the tumor. For example, here you have 3 copies from the low pass. And what you see in the whole exome data is that you have 2 bands at around 33 and 66%, because the 2 variant alleles from the germline SNPs, they will end up into the situation where you have 2 or 1 of those. And similarly, if you have this locus here, 2 copies, but LOH, what you can understand is that it's a combination of 2 events, loss of heterozygosity and gain of 1 allele. Whereas here you have 5 copies and you have 2 bands, as you can see from the local average, which is represented by this red dots, which represent the local average of the variant frequency. This corresponds to 40 and 60%, which sum up to 5 copies, 2 bands. So this all comes up nicely, and indeed we were able to validate, cross-validate the OncoSeek targeted panel. Covered region was very well reproducible also in the whole exome sequencing data in terms of zygosity and somatic mutation. So with this, I would like to conclude my part, and 2 main messages to take home. First of all, quality of the input sample for your downstream genetic analysis is key, and DEPArray is the technology to go for this kind of precision, and enables a very accurate characterization of copy numbers and sequence variants, even from very low tumor cellularity FFPE samples or from enriched blood. And purity is really, we feel, the key to make NGS regulatory friendly, because in this way, also the tumor samples become like of germline once you have them pure. And germline mutation is actually what the FDA and the other regulatory authorities are approving nowadays the NGS system, because then you have a clear understanding. Whereas if you go for somatic, then you have very high bars in terms of purity in order to have reliable results. And with this, I would hand it over to Matthew.
Hey, good morning or good afternoon, everyone. Hi, I'm Matt Moore. I'm principal and co-founder here of ResearchDX, and I'd like to thank Silicon Bio for the opportunity to present today. We are currently implementing the DEPArray technology in our clinical lab, and we're using it as a template selection device for use with our next-gen sequencing, qPCR, and FISH technologies. So really the question is, why would someone in a clinical lab want to validate another sample preparation device? There are absolutely many out there, so why would I want to do it? And really the answer is that when we look at our technologies, our current molecular assays, our qPCR, our microarray, our NGS assays, on the sample types we get, these assays are exquisitely sensitive and specific. However, the final performance characteristics of these assays are not so much determined by the instrument as they are by the quality of the sample input from clinical samples. And this is especially true for FFP tissue or CTCs. Clinical samples, we get all types. We get fine needle aspirates, we get large blocks with large proportions of tumor, we get old slides with small percentages of tumor, we get CTCs from three or four different purification technologies, and we're expected to be able to perform high quality analysis from all of these templates. So clearly from my perspective, the ability to isolate clean clinical clean populations of cells for use in the, in an NGS assay or qPCR or microarray assay is of great advantage from a clinical perspective. So what really does sorting using a DEPArray give us? It allows us to capture pure populations of cells, even from FFPE tissues, even from CTCs. These cells on the DEPArray device are captured in discrete bins, and I can retrieve these cells on a cell-by-cell basis, or I can put pure populations into a single bucket and retrieve multiple cells, up to 500 cells back at a single time. So from a clinical diagnostic perspective, what does that provide us? Well, it really provides higher sensitivity and lower costs for my NGS assays. While there is an additional upfront step in the purification process, I don't have to sequence it anywhere near the depth that I did previously. So no 10,000x depth is required anymore. I don't have to search for mutations in a 99% background of normal information that I'm not looking at. I can look at copy number and LOH from both CTCs and FFPE tissues that we could never do before. We can do copy number analysis from FFPE populations, which was just something that amazed me when I first saw it. More and more, we can even in populations, we can even look at clonal evolution. So the purity from an FFPE tissue allows us to do that. And mostly it provides us with the ability to separate tumor and normal populations from FFPE tissues. So we can look at germline mutations as well as somatic mutations in a very simple and clean manner from a single section. And finally, for assays that require whole cells, the disaggregation technology that provides enables us to do FISH on whole cells, whole disaggregated cells from FFPE tissues. And it's removing the sectioning artifact that you usually find with those tissues. So practically, how does this work? Well, you've heard a lot of it today, but from my perspective, the sort is achieved through an immunophenotypic characterization, an automated or semi-automated cell selection based on a number of physical characteristics. From a research perspective, there's many cool and interesting things you can do with this device in terms of separation. From a clinical perspective, we routinely use it to separate stromal tissue from tumor tissue. We can separate lymphocytes from tumor cells, and we can separate single or multiple cells even from FFPE tissues. It really is an enabling device from a clinical perspective. So how are we validating this device? That's actually a very interesting question. If you look at the CLIA regs from 88, hard-coded into those regs are the requirements for accuracy, precision, sensitivity, specificity, reportable range, etc. And these are absolute requirements. So how do we do it? Well, our current validation strategy is really a four-pronged attack. We're going to attempt to validate the DEPArray as a cell enrichment device, looking at the performance characteristics of the device itself. We're also validating the OncoSeek mutation panel, including CNVs from FFPE tissues. We have successfully validated a HER2 amplification assay by fluorescence in situ hybridization using enriched disaggregated whole tumor cells. And we're also validating their Ampli1 Low Pass CNV for analysis of single cell CTCs. So looking at how we're validating the assay as a sample preparation device, some simple experiments. We've taken FFPE sections from, dozens of tumors performed, standard IHC analysis, ran those sections through the device, isolated the tumors, isolated the tumor, isolated the normal, and then compared the bin cells back to the previous H&E and IHC analysis. And we have obtained 100% concordance of this. This device can absolutely separate, tumor cells, disaggregated tumor cells from disaggregated stromal cells and disaggregated lymphocytes, and with, a purity of, exceeding 99%, put into a tube for facial molecular analysis, only the tumor or the stromal or the lymphocytes, unquestionably. We've also tested, can these recovered cells be used for downstream analysis? We've performed stains on these bin cells. We've performed IHCs on these bin cells. We've fished them. In all cases, we can, these assays are technically correct in their performance characteristics. We have extracted both DNA and RNA in terms of nucleic acids. The quantity and purity of the nucleic acids produced is, equivalent or better than any other equivalent sample purification or collection device. And as was previously stated, the device actually can handle live cells, which in itself is an indicament of the way that it handles the cells through the process. So the third areas, are the molecular results of the cells concordant with results using conventional assays? We've looked at the OncoSeek CNV validation panel on FFP samples. We've taken FFP samples that have been validated or previously run in clinical tests, validated clinical tests. We've performed DEPArray sorting and compared the results and achieved 100% concordance. A couple of minor variations to be expected considering the increased purity from the DEPArray samples. We have successfully performed a HER2 FISH validation where we took a large cohort of both HER2 positive, HER2 equivocal, and HER2 negative cells, HER2 negative cases, which were previously run in other clinical labs, ran them through the DEPArray technology prior to fishing and achieved high concordance there. And we've also we're currently validating the Ampli1 Low Pass copy number analysis for single cell CTCs. That to me is a very interesting and highly promising technology. You can see concordance between each CTC. If you run multiple CTCs from the one patient, you can see concordances in copy number variation, but you can also see clonal differences, and you can see that in some cases a single patient will have 2 or 3 separate clones that you can verify using this technology and looking for the same result in multiple CTCs. Really is an extremely powerful technology. So the mutation panel, again, the validation set, we used a validation set from previously tested clinical assay results. We also created some sample analogs using known mutation status. So we created a bunch of FFPE cell line blocks with known mutation status. Mixed cell lines as well to make it a little bit more challenging for the technology. LOD, LOQ, with mixed samples at various concentrations, and unlike a traditional assay, the results all the way down to 5% tumor population are exactly identical, because the device, even if you only put a 5% tumor population in, you get 100% tumor population out of the device all the way down to 5%. So again, those results from a clinical perspective look extremely useful for us. Again, on our Ampli1 Low Pass copy number assay, The CTC samples, we've used previously tested CTCs that were previously tested using other technologies. And again, we have used spike sample analogs with known mutation and CNS status. And again, the results were concordant and as expected. The HER2 by FISH, there are actually guidelines in the validation of HER2 assays. We followed those guidelines with 40 positive and 40 negative samples. And again, we performed a precision study as well. Again, the concordance was, for all intensive purposes, 100% between doing normal FISH and FISH with the DEPArray as an enrichment device. So really, just to summarize, why are we validating these assays? Well, really, as I stated in the beginning, when you look at these technologies, when you look at the exquisite, sensitivity of an NGS assay, for instance, in most of these cases, the limitation of the technology or the limitation of the sensitivity of the output is a function of the population, of the template population that you put in, and this is especially true for FFPE tissues. And what we've found is that by providing an NGS assay with a small amount of nucleic acid from a pure population, you get far superior results than a large amount of nucleic acid containing that same amount of nucleic acid from that tumor population. So really the take-home message here is, your NGS data is better when you start with a small numerical amount of DNA from a pure population. Sorting cells for CTCs, there are plenty of bad CTC separation technologies out there which will reduce, which will concentrate your CTCs into 1 in 10,000 or 1 in 1,000. And the DEPArray is compatible with, I would imagine, all of those technologies. And the DEPArray enables you to take that purified that semi-purified population and really be able to analyze CTCs on a cell-by-cell basis. And finally, we used HER2 as almost a case study, but there are plenty of other FISH assays from FFPE tissues that could benefit by the removal of sectioning artifact. And we're actually, while we started on HER2, there's certainly a number of other assays that we want to implement to remove sectioning artifact by using whole sorted cells. And finally, why did we do this? We really wanted to demonstrate the utility of pure tumor populations in a clinical workflow, and we wanted to be able to provide this service to our clinical and our pharmaceutical customers. And with that, I would thank you very much for listening to me today, and I'd like to pass back to Faraday for some concluding remarks.
Thank you, Matt. So we have time to address some questions that were proposed by our guests this morning. The first is, does deparaffinization of FFPE tissue allow improved staining of tumor cells using antibodies that might not typically work in the immunohistochemistry lab? This is important for people who want to test the ability to identify specific cell populations using antibodies that may or may not work on standard paraffin-embedded sections. I would like to ask Nicolo if he can address this question, whether or not there's data to support the efficacy of deparaffinization and improved antibody utility. Nicolo?
Well, let's say improvement, we don't have a really comparative data. I would rather say that the condition may actually differ a bit. It's just a matter of finding the right, let's say, conditions for antigen retrieval. Of course, when we deparaffinize, we also disaggregate with some gentle enzymatic digestion. So I would say this is why also we developed the kit, because we wanted to provide a turnkey solution. But as with all other, let's say, immunohistochemistry, you need to find the proper conditions to make your antibodies work.
Okay, thank you. I think that was informative. The next, what's the average time to conduct an Ampli1 Low Pass tumor profile for CTCs? And are there any data demonstrating intra or inter-laboratory reproducibility for either this kit or any of the other kits? Nicolo, do you want to take that one again?
Yes, okay. So the protocol is very streamlined because you don't have to run all those, let's say, enzymatic digestions, fragmentations, and things. So starting from the WGA product, you basically have a simple PCR cleanup step and then pooling by the appropriate amount. So it's overall it's 2 hours hands-on, and a turnaround time of about 6 hours to prepare the library for sequencing. And then, of course, depends on the sequencer, whether the ion torrent you normally load it in the evening and you have the answer the next morning, or for Illumina, something similar. Although what we have today on the market is the ion torrent, but the Illumina protocol is quite similar. Basically, next day you get the result ready for bioinformatic analysis, which, by the way, is very, again, streamlined because of the specific properties of Ampli1, which make our whole genome amplification particularly apt to normalization, a very clean normalization. We didn't have to change the bioinformatic pipelines. We were able to go plug and play with existing open access software like Control Freak, which is widely available.
Thank you. That was a great result. Oh, go ahead, Matt.
Do you want to comment on the reproducibility?
Yes, I do actually.
We exchanged the samples before triggering you on the validation, as
right.
you remember.
So to answer your question in terms of, I can't comment in terms of between labs, but certainly in my own lab, you know, 2 sorts of repeatability and reproducibility that we've performed. If you look at multiple CTCs in the same run, you will see the same genetic profile in all of those CTCs every single time. And you know, there are cases that may have 2 independent clones, and then you'll see, you know, if you run multiple CTCs, you'll see multiple, you know, 2 distinct multiple copies of 2 distinct clonal populations. Now, if you run 2 separate experiments on different days or different weeks, again, we have seen really high reproducibility. The results will be concordant for all of the major, you know, structural differences that you observe. So I hope that answers your question. But yeah, in our hands, from our perspective, different cells within the same patient will, you know, give you the same result, and that same patient run on different days with different kits with different technologists will give you the same result.
Thank you, Matt. And then I think just one final question that I can address, is FFPE grounded before cell sorting? And the answer there is we do an enzymatic dissociation. The specimen is not mechanically grounded or displaced into single cells. It's enzymatically done. So with that, I'd like to conclude our webinar today. I'd like to thank our speakers, Nicolo Menarisi and Matthew Moore, as well as Shai for hosting this. And thank you all for joining us today.